I want to talk about synthetic biology. This is a reaction podcast to a conversation that James Collins had at the A16Z podcast about 3 years ago, so a while ago. It's about synthetic biology. James Collins is one of the pioneers. He calls himself a bioengineer. So what he really does, he kind of combines electrical engineering with biology. So for example, he builds circuits that are instead of transistors, he's using, let's say, E. coli bacteria, and they're kind of like switches, so on-off switches. So, for example, you could design a bacteria that emits a certain color if there's anthrax in the air. So things like that, and of course, this is just an idea, but the point here is that you can potentially design living things in a way that we design machines that have a certain outcome. And he talks in this podcast a lot about that. He talks about the science and the engineering and the difference between science and engineering. He talks about CRISPR, of course, because CRISPR is the ability to change the genome, which then gives the ability to transfer the newly, changed genome into protein space, and the proteins are the workhorses. So you could then have a biological system executing certain tasks that we are hoping it could execute for us. So there's sort of the barrier and the transition from biology and engineering. He talks a lot about how biologists are a little bit reluctant to embrace it, how physicists and engineers are not reluctant. Sounds very similar to other areas, such as, for example, economics. Whenever computation and software engineering come into place, then the existing domains get a little bit uncomfortable. But that's not the main point. I don't want to talk here about the evolution of science or anything like that. I really want to talk about the opportunity that we see at Orange Capital Partners, because we do have quite a sizable position in what we call the CRISPR basket, which encompasses things like synthetic biology, genomics sequencing, and then, of course, CRISPR technology. By the way, he also talks a lot about sort of the platform versus product company. So this is a very important podcast. Anybody who is interested in biology, SynBio, biotech, entrepreneurship should listen to this podcast. Again, James Collins on A16Z. It's about 2019-ish. There was a replay just recently. That's why I listened to it. And here in this particular episode, I want to sort of draw the line between that conversation and what we're doing here at Orange Capital Partners. This is Not Even Wrong podcast. It's July 7th, 2022. This is Krim Delco. For the year, the fund is down 40.5%. The market is down 17%. We're outperforming at 23.5%. And the main contributor recently has been sort of a macro revival. I think what's happening generally in macro is that oil and commodities are collapsing, and that is relieving sort of the expectation of inflation, and that is then also putting relief on the fear of further interest rate rises. And this is why the macro is more favorable right now for growth stocks. I think the big risk is, I would say, the dollar. The dollar is strengthening against the yen, against the euro. We believe that in itself is a good thing. I think that has been way overdue, the strengthening of the dollar, just based on economic performance and things like that. But the point is, of course, that when you have these types of moves in currency markets, you will have hedge funds collapsing, you will have arbitrageurs getting in trouble, and that will cause volatility in the market. So we have to expect that. But again, the fundamental sort of thesis here at Orange Capital Partners is to stick this through and just monitor the leverage, and that's what we've done in the last couple of months. So let's get to the conversation of synthetic biology. First, synthetic biology is the thing where you use engineering practices, such as, for example, electrical engineering, to create switches and circuits, but instead of having transistors, you're using biological systems. For example, bacteria. You can also use other organisms where you change the genome in such a way that then the proteins get expressed differently, and then the actual thing, let's say the bacteria, does certain things that you want it to do. Collins talks about the idea of having bacteria as sensors for things like anthrax, but this is just an idea. Of course, this can go much, much further. Now, what we think, we want to talk about two things fundamentally. First, a little bit about the history of synthetic biology. It goes back to the late 20th century with the Human Genome Project, and then continues until today. I want to talk about sort of the steps that were taken, and also the reason why some of these ideas have failed in the sort of in entrepreneur space, if you want. And then I want to talk about why we think today is different. So the history of synthetic biology, I think it goes back to like the late 20th century. The Human Genome Project is the big push into, okay, listen, we want to be able to read the genome. And it's not just that we want to know the human genome. We also want to know how to read genomes, and we want to understand genes. We want to know what is a gene and what is the function of a gene. So all these questions start, of course, much earlier, but then the technology and the money and the investment are there. And by the late 20th century, so sort of around the millennium, I think President Clinton produces the first human genome. Costs, I think, about $3 billion to read, very expensive. And this is the kickstart of what we believe is one of the biggest revolutions of the 21st century, which is actually digitizing life science through understanding the genome and then expressing the genome in protein space and then creating synthetic machines, biological machines. This is sort of the idea. Now let's get back to history. We have the first sort of wave in the early 21st century, so the 2000s, where because of high oil prices, high energy prices, people start really getting excited about creating organisms that can sort of mimic what nature is doing with oil and carbon. So instead of digging for oil, we can create fossil fuels out of algae, for example. We know Khosla is a big proponent of all this, and I think his fund and himself and his reputation take a big hit because pretty much all the investments he does fail. Gabo is one of the big investments he did, and then there's a couple of others I forgot. We were following the space, looking at it, not invested, because the fundamental problem with these things was also, according to James Collins, was that you can do it in the lab, but you can't scale it to mass production, and therefore it's bound to fail. So this was sort of the 2000s. We visited, for example, a DuPont plant in Knoxville, Tennessee. They were they bought that Danish company, Danisco, doing things with sugar, and then DuPont was hoping to use that technology, enzymes and all that kind of stuff, to produce synthetic oil or produce oil out of switchgrass and then all that kind of stuff. Well, it all just failed because, again, on the one hand, making fossil fuels out of biological systems is difficult. You need a lot of land, you need a lot of energy, you need a lot of technology, and it just didn't work. So now we come sort of in the second phase of this synthetic biology push, and this is more applications into life science and agriculture. So particularly life science has some good results, and I want to also include our portfolio here, the CRISPR stocks. There's some in vivo success with Intellia, where you change the genome of a person and then you can attack things like sickle cell disease. So this is where we are today, and it's fairly successful. I think the stocks have taken a massive hit recently, but if you look at, let's say, the market cap of Intellia, it's still about $5 billion, which is a success. And you have companies like Caribou, you have companies like Beam, and so on and so forth. I think one of the things that James talks about, which is very interesting also for us, is that typically what happens when you have scientific discoveries, that the academics that are involved in those discoveries, they want to build platform companies. This is, for example, exactly what happened with Caribou Bioscience. So Doudna and her people, they were instrumental in discovering CRISPR, and then they created Caribou. The idea was to use the technology to do many things, so create a platform. And then they spun out Intellia because Intellia was supposed to be the product company. So this is exactly what James is talking about. You have platform and product companies, and depending on which one, I guess it's probably just dependent on the entrepreneur and the sort of inclination. For example, I know Jennifer Doudna is very interested in building tools to reduce the cost, because her idea is that as a scientist and entrepreneur, the best impact she can have is to lower the cost of research and development, and then let many entrepreneurs and grad students and research labs just do the work, and then hopefully good things will happen. product companies are different. CRISPR Therapeutics is one of them. Beam is one of them. Intellia. They take the technology and they build certain therapeutics. For example, attack sickle cells. And one of the biggest pushes right now, as we speak, is the allogenic CAR-T cell design. So this, I would say, is the biggest application of synthetic biology as of yet. I've already coined allogenic CAR-T cells as sort of the penicillin of our day, meaning if it succeeds, it really, really can change humanity at a big scale. Because if we can, what are allogenic CAR-T cells? CAR-T cells are basically, think about it this way, you have your own immune system, and the immune system is supposed to attack cancer, but it doesn't. That's why we have cancer. And we can do two things. We can inject T cells that actually do fight cancer, and that is what CAR T cells are. So CAR T cells, I forgot what the acronym stands for, but think about it. You re-engineer the T cell in a way that it actually attacks the cancer. So, and by the way, you make sure that it only attacks the cancer. So it's not just that it attacks cells that are unwanted, it only attacks the cancer cells. So that way you reduce that, what we call chemotherapy side effects to a minimum. It almost is, it's just, it's like a, well, proverbial pill that you take or an injection that just kills whatever needs to be killed. Now, allogenic versus autologous, it simply means that instead of introducing your own T cells into your own body that are re-engineered to kill cancer, you take anybody's. So you take off-the-shelf T cells. What does that do? It reduces time to market, it reduces cost, and it increases efficacy, because you, instead of taking an old person, cancer patients, T cells that are already weakened, you're taking healthy T cells from somebody else and re-engineer them. You have to do two steps. One is you have to make sure that the immunology effects go away. So if you take third-person cells and inject them into somebody, then somebody's immune system will react to the foreign body, and therefore we have to circumvent that. And second, we have to again have a cell that attacks the cancer. Now, I don't want to go too much into immunotherapy, but the fundamental problem with immunotherapy is that our immune system gets tricked by the cancer, and we need to overcome that trick and make sure that our T cells attack the cancer. So having said all of that, I think we at Orange Capital Partners see this as one of the biggest applications. This is why we are still relatively large investors in this space, because if this works, we're talking about, I would say, without exaggeration, trillions of dollars of market and market cap potential. Of course, it's not just that it will create all this value. We have to make sure that we can capture the value. This is 0 to 1 Peter Thiel. You have, I think, what he calls A and B, or alpha and beta. So alpha is the value you create, and B is the value you capture. And that multiplication should be maximized as an investor. And of course, we want to make sure that happens. And it's a little bit difficult to make sure that will happen. So we could be still in the sort of first phase of allogenic CAR-T cells, and maybe the second, third phase will succeed. And we just have to make sure that even if we fail now, we keep we keep following the space, and we keep our feet close to the ground, so we are able to invest in hopefully the companies that will succeed. If the companies that we're invested right now don't succeed. So get back to James Collins's comments. So he talks about this. He talks about the allogenic CAR-T cell therapies, which he thinks are the sort of cutting edge applications of synthetic biology. I think there's a lot more to be said here. There's also synthetic biology in the diagnostic space for, let's say, just COVID. Twist Bioscience, one of our portfolio companies, is very instrumental in that space. We have synthetic biology, let's say, also in the form of, for example, DNA storage. So that's another application that is possible, very different. But the point here is, just to summarize this particular episode, we are talking about the ability to use living organisms to digitally manipulate them, the same way we manipulate, electrical engineering products, and to have certain outcomes that are desired. This is really what we're trying to do. And if we can do that in life science, we can attack medical problems, we can attack agricultural problems, energy problems. It is possible to do that. The problem with biology is, of course, it's too complex, and here I want to finish with Demis Hassabis. I already had a reaction podcast to his point. He said something very, very interesting. He said that he thinks that mathematics is a very good representation of Newtonian classical physics, and he thinks that AI is a very good representation of biology because it's very complex, and therefore logic and math, the way we know it today, is not good enough. But AI, sort of the fuzzy logic of neural nets and the learning through software 2.0, basically creating algorithms and then feeding data and then having the algorithms find solutions to problems, that is, according to them, is the best representation of biology. James touches upon this also. He talks about AI and the ability to use AI to to solve problems in biology. So we believe that this is a massive, massive opportunity. Again, we hope that Demis and Elon Musk somehow get together and talk to each other, because now this has nothing to do with life science. This is also material science. Could be very, very advanced very rapidly if we use AI and the approaches of DeepMind in the realm of material science. Now, DeepMind's already doing that, but we believe that a combination of, let's say, Tesla and DeepMind would accelerate that, because Tesla is at the forefront of actually building things, and DeepMind is more the theoretical AI part of it. So if we could combine that, I think we could have massive advances. The same applies to life sciences. There, DeepMind, of course, has made huge strides with AlphaFold. AlphaFold was not known by the time James Collins was giving this talk at A16Z. So this is a big, big step forward. We have genome space represented in protein space. We can understand both. We can manipulate both now, or at least we can understand that, let's say, if we have a certain genome, we know how the proteins will fold given that genome. And then if we change the genome through CRISPR, we see what the differences are and then what the different functionalities are. And from there, we can then learn what functionalities are good, bad, and how we can manipulate things. So this, in a nutshell, is the opportunity. We're very excited at Orange Capital Partners that this is happening. And one of the biggest lessons I want to give, and the purpose of this podcast is to give people a sense for what it feels like to run a hedge fund. When you see that science, engineering, technology are all converging towards sort of a very simple solution, which is we have a genome space, we can manipulate the genome, then we can understand through AlphaFold how this will be represented in protein space, and then knowing what proteins then will affect, let's say, in a body, we can manipulate those living organisms to have certain outcomes. This is a relatively simple work chain, value chain, that we understand, and where entrepreneurs can actually have impact, and where capital can have impact, and therefore wealth generation can happen. Thank you.